So today, I have the pleasure of talking with two friends from way, way back when we met in Colorado decades ago, actually in 1980s. And they are experts on talking tongues. So the University of Miami School of Medicine has an interest in people who can talk tongues. They're experts on taste and second chemo sensation in addition to olfaction. They're both professors of physiology and biophysics there. They've spent their careers studying taste from the tongue, and then mainly in the tongue, but a little bit up into the brain, and they know a lot about that. So they're going to tell us what they know. But before they start talking about the science of taste, it would be good for them to provide us with some background on where they grew up, how they got interested in science in general, then neuroscience, and then taste. So Nirupa, let's start with you.
Grow up? I didn't know I was supposed to have grown up. So
this is Nirupa Chaudhari and her husband Steve Roper. I guess I don't even think I gave your names. Go ahead, Nirupa.
Yeah, so I sort of grew up in a whole lot of places, military brat kind of thing. But I did undergraduate training in India. I studied development, and then I got my PhD at the University of South Carolina, actually looking at the evolution of DNA sequences using sea urchins, which have a fascinating, well-documented evolutionary history. And so they were a really good model for many years for understanding the concept of molecular clock, for example, for evolution. Came from some of those very early studies when people, including myself, were looking at DNA sequence evolution. And then, so I've kind of done a lot of different things. I then went on to a postdoc at the University of Colorado Medical School, where I was looking at transcriptional patterns in the brain as a whole, because at that time in the 1980s, early 1980s, those were some of the first studies where people started looking at transcriptional patterns and regulation.
Before there was, genomes had been sequenced, and before there were methods.
Before we knew anything, we knew very, very little about the genomes. But so anyway, so I had interests in genes and gene regulation, but then I got really interested in neuroscience and the physiology of neurons and things. And so I sort of did a diversion there for a while and studied ion channels and in excitable cells, and then sort of put it all together from about the early 90s. I started studying taste, and I've been there ever since.
And did you start in mud puppies? Is that right?
No.
Who was doing? I remember somebody was doing. Oh, that was Steve. He was doing mud puppy stuff.
Right, okay. Right. So I have a much less colorful background, you might say, more mundane. I grew up on the West Coast in Portland, Oregon, and went to public high school there. Then I went to college on the East Coast. I graduated from Harvard a few years ago. And from there, went to England to get a degree in physiology. Now, whereas Nirupa is a molecular biologist, I'm more of a cellular and a tissue level physiologist. And I got my PhD at University College London and returned back to Harvard to do my postdoc there at the Harvard Med School. And from there, went to Colorado, where I took my first position. In fact, that's where we met, at the University of Colorado Medical School.
So
And
you're putting electrodes in cells in the taste bud, and Nirupa's looking at the genes and molecular.
right, right. Well, it was an interesting evolution because at about the time that back then, the metabotropic glutamate receptors were just being cloned and identified. And since one of the fundamental tastes, and we might talk about this later, is a taste of glutamate, specifically monosodium glutamate, I talked to Nirupa and we got talking about, well, gee, could it be something like one of these glutamate receptors that have been cloned and identified in the brain? So that's how we started to get working from her molecular standpoint and me from sticking microelectrodes into taste cells. And you're right, it was on a mud puppy. And the reason for the mud puppy, very briefly, so to dispel any of the curious listeners, the mud puppy is a strange amphibian found in the Mississippi River area. It's a salamander that's neotenic, which means that, well, bottom line is the cells are enormous. It's polyploid animal, and all of its somatic cells, the muscle cells, many of the nerve cells, and specifically the taste cells are huge. And it was easy, relatively easy to stick a microelectrode into them at that time. And it was one of the first cases of good, secure intracellular recordings from these sensory taste cells. But I soon abandoned mud puppy and went on to mice because, quite honestly, there was not a great deal of interest in learning how and what mud puppies respond to taste-wise.
And then, so since there's two of you, we'll have like a tag team question and answer. So what? Let's start out. What is a taste bud, Steve?
Sure. Okay. Well, I'm going to talk mainly, in fact, exclusive about taste buds in mammals and specifically taste buds in us humans. And the way to think about it, the taste bud, first of all, it's a sensory organ. It's a sensory organ for the sensations of gustation, taste. And a taste bud, each taste bud is like a small island of cells in the shape of like an onion or a garlic, this little oval, tight, compact island of cells embedded in an ocean of surrounding non-sensory cells in our oral epithelium, the tongue, the cheeks, etc. There are taste buds occurring elsewhere, but that's the primary site. So each taste bud, and there are what, about 5,000 to 10,000 taste buds total in a human on average, although some humans curiously have maybe twice that many more taste buds. And they tend to be females that have these greater number of taste buds, that I understand. And those individuals really have a heightened sense of taste in many ways. It's like it's been said, it's like neon colored taste to us normal taste people.
So this may be how, yeah, so women should be better food tasters. Like if you're going to be a restaurant critic or something, it might be better off being a female.
Why is that?
Well,
because you have more taste buds.
Well, I exaggerate. It's only a small proportion of the population, and that portion has a greater percentage of females, but you cannot say that women have.
No, I'm joking.
Of course. Although it's clearly many women have better taste than.
Yeah. So. That extends beyond the taste buds.
Exactly.
Okay, so then Nirupa, so within a taste bud, are all the cells the same, or are there different kinds of taste cells?
Right. So, well, okay, so they're not all the same, bottom line, to your question. But when you say how many types of cells, it sort of depends on what one is looking for, right? Just like if you said how many types of people are there. So are you looking for short and tall people? short versus tall people and everything in between? Are you looking for really smart versus really artistic? There's lots of different ways to categorize. And if you use just morphological criteria, shape and size, then you can define some cells that sit near the bottom of the taste bud, and they tend to be the cells that are just precursors. They're incipient cells. They're little babies that are sitting there waiting to become real live functional taste cells.
So they're stem cells?
The stem cells are not inside the taste bud. They're sitting on the outside in the surrounding basal layer of the epithelium, by and large.
Oh, I see.
But there will be, so once they have undergone their final mitosis, they've crept into the inside of the taste bud, but they don't differentiate until there's a need, because there's a sort of a static size to a particular taste bud. And so, when one of the cells dies, and there's continuous turnover, the cells are not very long-lived, depending on the cell. It's a few days up to a couple of weeks, maybe a couple months at the very most. So then they'll regenerate from these basal cells. Then there are cells that can detect sour substances, and they have some specialized properties. They're the most neuron-like. They're excitable. They have voltage-gated calcium channels. They have big and clear synaptic structures, things like that. And then there are cells that are really receptors that detect. So they have the G protein coupled receptors for detecting sweet or bitter. So the cells that detect sweet are typically different than the cells that detect bitter. But other than those molecular receptors, other features are similar. So there's some convergence and cohesion between the morphological characterization and the functional characterization. And in fact, over the last 15 years or so, the field has finally sort of come to a good convergence, because initially, of course, the definition was based on electron microscopy. And people spent a lot of time arguing about, well, it's this cell type that does this, but in fact, we didn't have a clear correlation between function and morphology.
And so you mentioned kind of three different categories of taste: sour, sweet, and bitter. And what about, there's also salt? In cooking, people use salt a lot, and then
Sure.
And then there's a fifth one.
Right, umami.
Yeah.
Umami. Is that what you want to talk about?
Well, salt and
Salt
umami.
is a specialized cell type as well.
Okay.
And it may be more than one cell type that detects salt, and that's a little bit still up in the air, not completely worked out. There's a cell that detects salt under some certain conditions, and there may be, but then salt itself is many things, right? So we think of sodium chloride, which is the essential salt that we need, right? And we always need sodium chloride. But then there are lots of other inorganic salts that you can taste, or organic anion salts. And so that's where things get complicated with salty, because we don't have a tight handle on what is the commonality. Are all sodium salts detected just for the sodium part, which is probably what happens. And then the anion part is sort of really somewhat mysterious, and we don't know exactly which cell type. There's clearly a difference between different salts.
Yeah, that would seem kind of a, because you got sodium, which is an excitable, so sodium's higher concentration outside the cell than inside, and excitable cells, when they depolarize, the sodium goes in. So like how much of an increase above the kind of basal sodium level does it need to activate these?
Right. So that's interesting. As a physiologist, you're asking a physiology question, and that's actually something that people had recognized for a long time. But here's a little fly in the ointment of that argument, which makes it even more complicated. So normally, if you think about a neuron, it has low sodium on the inside and high sodium on the outside, like your tissue fluid is 140 millimolar sodium, and there's just 10 millimolar on the inside, so sodium's wanting to go in, but it's kept out, all of that. But then, if you look at the cells of the taste buds, most of the area of the cell around the belly of the cell is surrounded by that tissue fluid. But the business end of the taste bud cell is out in the oral cavity. And saliva doesn't have a lot of sodium in it.
Oh, I see.
So you see, what happens is that, in fact, on a baseline of about 10 millimolar sodium, something like that,
I see.
if you just go up to 30 or 40 millimolar sodium, you can detect it.
Okay.
Yeah.
Interesting.
So if I could jump in here again, just getting back to your question, though, I think for our listeners, make it clear that there are not simply 5 different types of taste bud cells, one that responds to sodium, another responds to sweet, another responds to bitter, etc. In general, you can say there are 3 broad classes based upon morphology: a receptor sensory cell, there is a glial supporting cell that we haven't mentioned yet, and there's a basal cell. Now, of the receptor cells, in general, there are some that respond primarily to sweet and others that respond primarily to bitter. But there are some cells, taste cells, receptor cells, that respond to multiple taste stimuli. So it becomes a little unclear. It's not a sharp delineation of cell types. And those different cell types are currently being worked out in great detail by a number of laboratories.
I see. Okay. So umami taste is, so my understanding is monosodium glutamate is like the classic umami taste. Is that right? So we know that glutamate in the brain is the most prominent neurotransmitter. It's excitatory. Are these receptors glutamate receptor? They're type of glutamate receptor?
Okay, so in our initial study, we'd identified something which was a modified brain-like glutamate receptor that probably has some kind of role, although we weren't able to exactly define exactly what that role was for the metabotropic glutamate receptor that was in the taste buds. And there seems to be some variation species to species, etc. However, there is also a different G protein coupled receptor, which is not related to or not closely related to the metabotropic glutamate receptors in the brain. And it's much more related. There's a class of G protein coupled receptors that are called the taste receptors. And there's a dimer of those that also responds to monosodium glutamate, to glutamate, and also, depending on the species, to other amino acids, and it's really highly heterogeneous. And so some species detect many different amino acids, and then some species detect glutamate in preference over a lot of others. But glutamate is a very common amino acid, right? And so if you want an indicator for protein, it's as good as any.
Yeah. All right. So Steve, you have graduate students, postdocs in your lab. They're putting electrode in these taste cells, and then in some say mouse tongue preparation or something like that, and then sort of recording, you add a specific tastant. You know, it could be sugar, it could be glutamate, it could be something with a bitter taste. Caffeine, for example, has a bitter taste. And then can you, when you're doing this, you can distinguish which of these, you know, subtypes of taste cell you're recording from?
Oh, yeah, certainly. Although
Okay.
we use a slightly different methodology, but it's similar to intracellular recording that you just mentioned. Few people are doing intracellular or patch recordings on taste bud cells in a taste bud because they're very difficult to get to with your microelectrode. You have to push through a lot of tissue, and it's not very amenable to patch clamp recording. So what a number of us have turned to is to use calcium imaging or functional imaging. Not that it matters. The end result is, can we see the activity of individual cells? And can we identify a cell that responds to, let's say, caffeine in coffee versus sugar in a chocolate bar? Not that we use those chocolate bars as stimulus. And the answer is yes, of course, we can see those cells.
Yeah.
And with transgenic labels, you could tell which cell is which.
Yeah.
I see. Yeah, that's interesting. When we were at Colorado State University in Fort Collins together, it was a time when Roger Chen, who he passed away, but he got Nobel Prize for developing these certain fluorescent molecules that respond to calcium that you could introduce into neurons. And as you know, me and others started using that in Cater's.
Absolutely, yeah.
Yeah. And it's
we followed hot on your heels.
Well, I don't know about that, but
we're mostly using genetically encoded calcium.
Yeah, I know that. I know it's advanced and they're much nicer in many ways to use. Speaking on, you know, so there you got to use molecular genetic tools. You got to introduce a gene encoding this calcium sensitive fluorescent protein or whatever. So I'm talking about genetics. So have studies been done, you know, for a long time now, people have been able to create what are called gene knockout mice, where you selectively disable a gene so that the protein encoded by that, just by that one gene, is lacking in the mouse. Have people made knockout mice for presumptive receptors for these different tastants, and then like whatever, you know, for example, it seems to me if you knocked out the receptor for sweet taste, that could be bad because sweet means calories usually. And if you can't detect sweet, you may not eat as much. So I guess, can you elaborate on that a little?
Yeah, let me start, but of course the molecular biologist, my colleague here, should answer the full. But that's a very important point, is that the canonical way of establishing whether a receptor was a taste receptor was just as you described, to knock it out in genetically manipulated mice. And indeed, that has been done for the sweet receptor, and you can generate mice that do not sense their taste blind to sweet. There's some complications there, but we don't need to get into the complications. There are some that are taste blind to bitter. There are some that are taste blind to umami, and that gets back to our original question or discussion briefly about umami receptors. Nirupa, in fact, was one of the first to identify a taste receptor. It was an umami receptor, but there are apparently several receptors, taste receptors for umami. Nirupa had identified one of the first ones in that 2000 paper in the Journal of Nature Neuroscience. But we could not knock out that receptor because it was a derivative from a brain receptor. And if you knocked it out, you would eliminate that receptor throughout the animal, including the brain. So if you saw a change in the behavior of the animal, a change in its behavior towards glutamate taste, you could not really say with confidence that it was only in the taste bud. Now, subsequently, taste receptors for other taste receptors for umami were identified. They were able to knock them out, and that had a major impact on behavior. That mice became relatively taste blind to umami taste. So yes, it's possible to knock out these taste receptors. And you know, the curious thing is, and maybe Nirupa can comment on this, you knock out these receptors in mice, they live a perfectly normal, happy life, eating, you know, Purina rat chow or Purina mouse chow. They don't seem to mind an absence of sweet receptor or bitter receptor.
Interesting.
So remember, if you want to talk about sort of the evolutionary significance of taste receptors and particularly detecting particular tastes, the mice in the lab aren't selected for their ability to do anything, right? I mean, they have all of their needs taken care of. They live in a clean environment. They get all the food and water that they need. They get to mate if you should so choose. And so the lack of a particular function, the loss of a particular function sometimes in lab mice, if it's profound, you can make an interpretation and a conclusion out of it. But a lot of times, if it's something like, what is the importance of the sweet receptor for the life of the mouse, you're not going to get an answer to that when the life of the mouse is really in a very unnatural setting where they're living in a cage with everything safe, right? So, but if you talk about an evolutionary perspective, people have always said, well, sweet taste evolved in order that we or any animal should be able to find calories, you know, and sugar, sweetened fruits and things. Well, that's all true, but in fact, a very common source of calories and carbohydrates for most animals is not fruit, but complex carbohydrates. Whether you are a grass-eating animal, a leaf-eating animal, a grain-eating animal, none of those are really sweet. And so complex carbohydrates are not sweet. It's just the simple carbohydrates that are. Similarly, umami as an indicator for protein works to some extent, but then you realize most amino acids are encountered as polymers called proteins, and that's what your nutritional need is. And proteins don't have a unitary taste quality to them at all. And so, so the evolutionary argument is it sort of goes only so far. I think it sort of makes sense, but you know, so you can have indicator tastes, just like, you know, a monkey may have developed its, you know, sort of scotopic vision and, you know, really focused on red because those were the fruits in the trees kind of thing, that the argument that you hear. But then not every red item is a fruit to eat. So, you know, I mean, the animals use what
seemed like, so bitter taste, you know, usually, and I actually wrote a Scientific American article on this in 2015 that with this kind of the concept that many of the chemicals in, say, the skins of fruits or in vegetables that are good for our health, the reason they're actually in the plants is they act as natural pesticides to keep herbivores and us from eating too much of them, but we co-evolved with them. Right. And, you know, bitter tasting, you know, plant materials, they often have other nutritional values. So anyway, so for example, I mentioned caffeine. If you put coffee beans or tea leaves on your kitchen table in the summer, you're not going to get ants attracted to them, right? They kind of avoid them, whereas you put sugar. So, Yeah, it seems to me the bitter tasting in particular from an evolutionary perspective might initially been to keep the animal from one eating the plant, but actually
Right.
having a toxic detrimental effect on them.
So yeah, so again, many toxins are bitter, and so they're naturally avoided. But then many beneficial compounds that are bitter are in fact consumed quite regularly. So it's just an extension of the same thing that I was saying, is that the taste indicators, and then there's learned preference for particular tastes as well. And so people will seek out coffee because you like the effect that it produces on you. And so that's yet another, it's a behaviorally adjusted preference.
Yeah, that's interesting point, that over time you, like beer, for example, probably
Exactly.
the first time I remember, the first time I drank beer when I can't remember, probably underage. It was it didn't taste that well, but you drink a little more and then you start to like it. Same with coffee and tea. So there's got to be some so maybe that gets us into the coding of taste, because we eat foods and we can close our eyes and somebody give us some food and we can oftentimes say exactly what food that is, but that, so that indicates it's actually complicated. It's not like this one chemical in the food is activating one type of taste receptor on one taste cell, and then that signal somehow goes to a region in the brain where that chemical's taste is stored. So how does this coding work? And maybe both of you can talk about this, because it's complex. I don't understand it. I'm not sure if it's well understood, but there's kind of some different theories of, like I was reading actually your review articles, I think, and there's this label line coding model and the combinatorial coding model. So
you could do this.
Yeah. Okay, so Matt, let me start on it, because in fact, just yesterday, I delivered a lecture to our medical students here on that specific question, not in taste, but sensory coding. How does information from the outside get to our sensory receptors? What do the how do the sensory receptors transduce that into an electrical signal, and what does that electrical signal mean to various centers of the brain, as you sort of alluded to? The short version is that there's not, when we say something tastes of a certain quality, like tastes like strawberries, there are a whole host of sensory information bits that come in. There's the sugar taste of the strawberry. There's the color of the strawberry. Color is very important in identifying foods. There's the fragrance, the odor, and that turns out to be absolutely essential for identifying a food. And what people commonly call taste is actually flavor, the mixture of olfaction and gustation. And indeed, probably the olfactory component is even more prominent and important in identifying food substances. So we've got the gustatory taste input, we've got the olfactory input, we've got the color, we've got tactile, the texture, the difference between a wet potato chip and a fresh potato chip, absolutely astounding difference. It will help you identify. There's temperature. So in short, taste, when people say, oh, this tastes good, or this tastes like such and such, it's really this symphony of sensory input that comes in. And you have to take that into consideration when you consider coding in the brain, because all of those sensory inputs have different routes into the brain. Most of them go through the thalamus and then up to the cortex and then to association cortices, etc. So it's a very complex answer, is that there's no simple way to describe coding. Now, having said that, we can, of course, identify neuroanatomical pathways for the gustatory inputs and how they get to the brain, and how the olfactory inputs get to the brain, and where they interact. But, oh, by the way, an interesting thing about the olfactory input, it's not necessarily what's called orthonasal olfaction. When you breathe through your nose, it smells like. It's the food inside your oral cavity that generates what's called retrograde, or retronasal, rather,
Oh, interesting.
olfaction. And they're not identical. They're not identical. The retronasal and the orthonasal can activate different pathways. But short version, the retronasal olfaction and the gustation determine what we identify as a food taste. So I'll turn it over to just a second. Wait about it. So what else can I say about that?
Do you want to say anything about the label line coding and the combinatorial?
Well, okay. Yeah, the labeled line coding is a type of sensory coding that has been identified in insects for certain, and in lower animals, it's the concept that you have a single type of a taste receptor cell, which I've already told you, you cannot say that there's a single type, but that's the what you posit for labeled line. So let's say there's a sweet sensing taste receptor cell that makes a synaptic contact, and contacts a nerve axon that goes into your hindbrain, and that nerve fiber in the hindbrain only responds to sugar, and then it communicates to the thalamus, and the nerve cell in the thalamus only responds to sugar. It's labeled. This whole line is labeled all the way up to the cortex, and somewhere in your cortex there is a cell or a cluster of cells that respond only to sugar. It's labeled, and that would identify sweet. This has been a very attractive concept because of its simplicity, and there have been attempts to identify these labeled lines, and some people have said, yes, they've identified them, but more recent studies and more accurate and comprehensive studies have clearly shown that this is way too simplistic, that there's not a simple, consistent labeled line for sweet and for sour and for salty and bitter, etc. Now, the other concept is a little bit difficult to describe without slides or a blackboard. I have a blackboard here, by the way, but don't worry, I'm not going to go to it. I'll try to paint you a word picture. It's where you have multiple inputs coming in, and it's like a population votes on a taste, and even though each individual member of that population may respond slightly differently to the taste compound, some predominantly sweet but with a little bitter, others a little bit of bitter and a little bit of sour, but when you add them all together and you combine them in some sort of complex way, the output generates the perception of sweet. Now, where this combination takes place is probably throughout the taste axis all along the way. So that's sort of a word painting of what combinatorial coding might be. But in addition, even more recently, there's been a lot of evidence that it's not just the combination, but the timing of how they combine. It's not only a symphony of taste coming in together, but it's a rhythm at which they come in together, and the rhythm of the firing of the action potentials.
And then
it becomes the bottom line here that I'd like to convince you is that the labeled line concept is way too simple. It may work for flies, and it may work for C. elegans, but it most definitely does not work for humans.
I see.
Or other mammals.
Yeah.
And so in order, obviously, as with all your sensory input, there's some convergence of inputs. So in the case of food, right? So this information ultimately is ending up, say, in the hippocampus, which is very important for the short, initial establishment of memories. And then somehow long-term memories may be encoded there elsewhere in the cortex. So, simultaneously, you've got you eat some food, you have some taste cells being activated, some olfactory neurons being activated, then, those signals are going back, and somewhere they're converging in time and probably space, that is in synaptic, simultaneous synaptic. As you know, I used to work with single cells a lot in culture, and so I think of like, okay, you've got one axon coming in from an olfactory stimulus into a dendrite of a hippocampal neuron, and there's another axon that's coming from information come from a taste receptor, and it's synapsing on that same dendrite. And so when they're activated at the same time, you get a bigger calcium elevation and a strengthening of those synapses. But anyway, so I guess I'm not asking a question more than just kind of making a point that.
Yeah.
Well, let me help clarify that just a little bit, Matt, because you focused on the hippocampus, which is absolutely true for our long-term recollection and memory of taste, but probably what you're defining is the interaction of olfaction, gustation, mechanosensation, temperature, probably first in an area of the brain called the insular cortex. It's a sensory cortex, primary cortex for taste, and a lot of interaction, this combinatorial coding that I'm talking about in your description of a convergence of inputs probably occurs in that insular cortex, and then from there, the information goes to other regions, accessory cortices such as orbital frontal cortex, but probably where that initial main interaction and coordination occurs is in the insula.
And in humans, so I presume there have been functional magnetic resonance imaging studies where someone, you can image the insula or whatever, brain regions, and have someone taste something or eat something and then see which pathways. So Nirupa, is that been done, some of that?
It has. It's been done in rodents. It's been done in people. And one of the problems is that the insula is not an enormous area.
Right.
Okay. And so if, in fact, there are very fine subdivisions and things like that, and exactly pinpointing this happens here, that functional MRI may not. I think that you can still sort of say, oh, but there are clear spots where only sweet taste appears. And electrophysiological studies, which of course have much higher spatial resolution, which have been conducted in monkeys as well as in rodents, pretty much say that the coding aspects, that they're distributed across these cortices.
I see.
Okay? And so you have convergence of somatosensory from trigeminal, olfactory, and gustatory input, and these convergences and recurrent circuits actually occur between the olfactory cortex, piriform, and the gustatory cortex. They sort of go back and forth, and so taste will inform olfaction, and vice versa. Olfaction informs taste, and so those circuits are actually fairly complex loops with a lot of information going back and forth. It can be demonstrated. This sort of integration can be demonstrated with very simple behavioral assays. For example, it's something that was done a long time ago, which is that you can have somebody test the threshold for sweet, right? So you have sweet solutions that are progressively diluted. And if I give you a drop of this, you can say, oh yeah, that's sweet. And so until it's diluted enough that you don't detect it as sweet. And then you can have odors. And if I give you, find this threshold below which you cannot detect strawberry odor, and then combine that subthreshold concentration of strawberry odor with subthreshold concentration of sucrose solution, all of a sudden you can perceive strawberry.
Huh. Interesting.
It's absolutely, so neither one by itself tells you sweet or strawberry, but the combination tells you. And that is a very clear demonstration of the integration that occurs across these senses.
Yeah, that's fair. That's nice. Those are nice experiments.
Yeah.
Very interesting.
A different experiment that I actually I didn't conduct this, but I was a subject in one of these, and it's really fascinating that you don't identify foods very well at all. Even really, really common things, even things that you know you love, you don't identify them outside the context in which you know them. And this was actually really, whether it's intact foods or whether it's just a smell. I mean, I was just completely amazed when an otolaryngologist researcher who I knew said, okay, let's see how well you can smell and identify. And he presented me with these little vials with odors, just odors in little vials. And so there was no context, there was no color, there was no, oh, here's a pineapple. I couldn't identify pineapple.
Huh.
I mean, like I couldn't put a name on that. And I go, yes, this is a fruit. I know I like this stuff. And I couldn't put a name on it because there was no context. But the experiment was with Jell-O. And when they took cherry Jell-O and doctored the colors so that it was no longer red and gave you a cube of it, you couldn't tell it was cherry Jell-O.
Wow. That's
yeah.
And one of the practical things about this, Matt, is from a clinical point of view, is that patients, and for example, in the COVID pandemic most recently, will go to their physician and say, Doc, I can't taste any longer. And the problem is it's probably not taste that's gone wrong. It's most likely olfaction or some other sensory mechanism. But this confounding of all the senses and calling it taste can be very, it's incorrect and misleading.
Yeah.
Well,
it's just
as long as you mentioned COVID, we can talk about that. So, yeah, so sense of smell and taste, which could be secondary to that sense of smell. So you mentioned early on in our conversation that, I think Nirupa mentioned this, that these taste cells, the sensory cells, and this is true in the olfactory system too, they're constantly turning over. You know, some of them are dying and then being replaced by cells derived from stem cells there. And so what's the thinking with, or do you know, with COVID, is it somehow the virus killing? You know, we know it gets in the, one of the main routes of entry is the nose, right? Is it actually infecting and killing the cells, or is it just like diminishing the function somehow?
Right. So for olfaction, what happens is the virus, the SARS-CoV-2 virus, actually infects not the olfactory neurons. It infects the sustentacular cells that are supporting the olfactory neurons. And so
sorry, are those like glial cells?
they're epithelial cells. So the nasal epithelium, the olfactory epithelium is they're tall, they're columnar cells, and they basically encase all of the olfactory neurons in that epithelium, because the neurons, of course, also span that whole epithelium. with the dendrites out in the outside nasal space. And so when the sustentacular cells get infected, then that actually has a secondary effect on the neurons. So initially it was thought, well, perhaps the sustentacular cells are just sort of falling apart, and then that secondarily causes the neurons to disappear because they need the support from the physical as well as metabolic support from the sustentacular cells. It turns out actually that the neurons, even when they're there and viable, they're actually downregulating the expression of the olfactory receptors, which is really quite amazing. And so even in a mild case, the olfactory receptor gene expression gets downregulated. And that seems to be sort of part of the loss of the olfaction, and then potentially the parosmia, where things come back, but they come back scrambled, etc.
I see.
In taste, it's a little bit different, which is that, as Steve was saying, there's this general popular confusion or conflation, really, between taste and smell. And so people will say, even with a cold, people will say, I can't taste anything. But there's absolutely nothing wrong with your taste when you have a cold. It's that your nasal passages are blocked, so you can't smell. And as we were just saying, a huge part of the recognition and enjoyment of food is from that integrated sensory signal. And so if you lose the olfactory component, all of a sudden it feels like you've lost all of taste.
Yeah.
But if it's tested in isolation, people can taste just fine. They can tell this is sweet and the other thing is bitter. But apart from that, in COVID, there seems to be some loss of taste genuinely, apart from this conflation that I was just mentioning. And we don't have a real clear mechanism for how that happens. It could be literally something as broad and general as an inflammatory change that sort of influences neural conduction, for example.
I see.
Yeah, so we really don't have a handle on a complete mechanism for why there would be loss of gustation. But the good news is that the more recent variants of the SARS-CoV-2 virus aren't exhibiting as much of that olfactory and gustatory exhibit. The initial couple of variants were the biggest culprits of all. And we're seeing much less of that now. So that's good.
Interesting.
And I think it's fair to say that the loss of gustation and the loss of taste per se in COVID is much rarer, much less common than the loss of olfaction. So
I see.
Even though people say they don't taste things, the loss of taste per se is pretty insignificant, I think.
Okay. Kind of switch topic a little bit. So when we, people our age, when we grew up, so I was in high school in the early 70s, and, thinking back on it, I don't remember many kids in my classmates. I think I can remember one that was, clearly overweight. Right. And now it's very different. There's a lot of overweight people. So we have this obesity epidemic, and there's a lot of factors involved, but one is that people eating a lot of fast food, junk food, maybe, fructose, simple sugars. And these, my take on the processed food and fast food industry is obvious. They want to get their people addicted to the food so that they come back to make more money. So when I was at the NIH, so I was at the National Institute on Aging, and our research labs were right next to the National Institute on Drug Abuse. And I knew Nora Volkow, who's the director, and she's done a lot of work on food addiction and, showing that, I guess it's kind of the way that you give somebody a Big Mac, and the dopaminergic neural circuits involved in reward and pleasurable experiences light up. Is there any studies looking at the dopaminergic system when, I guess, an animal or maybe even human is experiencing some taste? Say, let's keep it simple, sugar.
So, yes. So I think that the activation of dopamine and some opiate pathways, right, has been demonstrated. The thing with addiction is a little bit kind of farther afield, because we derive pleasure from many things in life, right? You see an old friend, you smell something nice, you even just remembering something. So we think that there are reward pathways that get activated in the course of daily life, and that they help sort of supplement and enhance experiences in a variety of ways, right? So saying that a dopamine pathway is activated doesn't mean that you're on the path to addiction, right?
No.
So the thing with the addiction piece is the part that's really sort of unsettled, perhaps, if you will, or that there's not a very clear consensus about food addiction per se, I would say.
Just from kind of a simplistic thinking, it's
right.
People continue doing something that makes them feel good, even though they know that it's in the long run not good for them.
Right. So in fact, people have used the sort of the formal psychiatric diagnostic definitions and sort of mapped eating behaviors onto these and come up with something called an addiction scale. There's something called the Yale addiction scale, food addiction scale, and and there are a couple of other measures that people do use to define these sorts of behaviors that involve excessive overeating of particular foods and what are the drivers for these. And it's, as you said, it tends to be the hyper processed and hyper seasoned, a lot of salt, a lot of sugar, a lot of fat, some combinations of those. And so I think part of the problem, though, is that addiction, obesity, overeating, they don't all exactly sit on top of each other. And so people have tried to do fMRI studies, I think, but it's, defining the different categories and how you place different individuals in these categories, I think is.
Yeah. And of course, from evolutionary perspective, it's a good thing to eat a lot of food when you have a chance to, because our ancestors and animals evolved in environments where food is scarce. So it's, our whole system's geared in a sense to overeating because, give us increased fat stores that, maybe we're not going to get any food for a couple of weeks or a month. So, but okay, we're getting off topic a little bit. Before we finish up, from a science standpoint, what fascinates me is these the taste cells seem to produce a lot of proteins, peptides that are involved in regulation of energy metabolism. And actually I was reading a review article. It's actually by Josephine Egan,
Oh,
a
yes.
colleague, and so apparently there's insulin, somehow insulin released, and it can cause, somehow when, I guess it's sweet taste cells are activated, then there's a relatively rapid increase in insulin release from the pancreas that
Yes.
occurs even before, that sugar is getting into our intestine, into our system. You want to talk a little bit about some of the
Sure.
proteins? Yeah.
Sure. So, okay. So that I'm of course familiar with Dr. Egan's work. It's a little bit complicated. So there are receptors on taste bud cells for a number of these metabolic regulatory proteins and things. And I think that the evidence for release of peptides from taste buds or of modulation of taste bud function by these peptides is sort of wobbly at best, I'd say. But on the other hand, what is very, very clear is that that insulin is released very rapidly from the pancreas very soon after you taste sweet. And this occurs in rodents, it occurs in people. Some of the specific parameters of which sugars or synthetic sweeteners will trigger that, that's a little bit still up in the air. There's some conflicting evidence from different species, et cetera, so we won't go into all those nuances. But the concept is simply that if it's a sort of a call out, it's a shout out, oh, sweet coming down the pipe. So this is long before glucose levels in the blood go up. So the pancreas doesn't have a direct read on blood sugar. It's getting a neural signal, which likely goes from the taste buds into the brainstem in the nucleus of the solitary tract, and then probably via the vagus activates vagal and sympathetic and so autonomic neurons would then be activating that small release. It's a very small release of insulin compared to the much larger release that occurs when blood sugar goes up. And it's not just insulin. There are lipases that similarly are triggered to be secreted into the intestine when a lot of fat is tasted. So these may be sort of just early callouts.
Yeah, that's a good way of looking at it. It's like preparing the whole system for what's coming down the tube.
Right. That's what, and in fact, it's been demonstrated that you don't even have to swallow sweet taste. If you just have the sweet taste and paint it onto the tongue, but you don't swallow, so there's no glucose to increase in your bloodstream, you'll still get this what's called the cephalic phase, or this reflex increase in insulin. Small, but as Nirupa said, it's to prepare the digestive system for food coming down. But what Dr. Egan has gone on to show and believes that the taste buds themselves generate and secrete insulin, a small amount of insulin, and I still think that's debatable
and its significance isn't clear at all yet.
Right.
Right. So we're almost done. This gets the question. So you've been talking about one end of the food tube, right? The first thing that, and it's fascinating that it turns out, like I've seen a couple of papers suggesting there are taste buds in the intestines. So what's kind of the current thinking on similarities between cells in the epithelium in the gut, lining of the gut, and cells up in your oral cavity?
Let me start on this one. Well, first of all, yes and no. There are not taste buds. Remember, the taste bud, as I hope to convey at the beginning, is a compact little cluster of sensory receptor cells. Those exist mainly in the oral cavity and in the pharynx area. But there are isolated receptor cells, and more importantly, that there are non-taste cells that express the G protein coupled so-called taste receptors throughout the body. They're in our brain, they're in our GI tract, in the gut, they're in the airway system, they're even in the urethra and in the testes. These taste receptors, so-called taste receptors, are expressed in many, many regions throughout the body. And they obviously play different roles. For example, in the airway epithelium, the bitter taste receptors that are expressed in our trachea and the airway epithelium respond to bacterial exudates and they sense infection, bacterial infection, and cause the airway epithelium to start, the cilia to start moving more rapidly and to try to exclude those bacteria. So that might be what you've heard about in the expression, the appearance of taste buds throughout the body. It's not really taste buds. It's a receptor.
Yeah. So it's just those G protein coupled receptors that are used for detecting bitter compounds, and they're called taste receptors because that's where they were first cloned from.
Yeah.
But it turns out that that these bitter taste receptors are expressed in a whole lot of different places, and they serve a variety of defensive functions in a whole lot of these other locations, detecting perhaps metabolites that are warning signals. They might be detecting pathogens that, again, would be warning signals. So the bitter taste receptors, just like they reject bitter compounds in your mouth, are in fact sentinels for deleterious compounds, metabolites in several different other tissues of your body.
And has this been in mice that lack those receptors, the bitter taste receptors? You'd think they have a lot of different phenotypic changes when they're. This is something I've found throughout my career, that if you study something in like a basal unperturbed condition, you might not see much, but if you put some environmental challenge on it, then you see something.
So in fact, it turns out, I can give you an even better example than mice, but people have really, there are now studies, population studies with decent numbers in them that demonstrate, for example, that polymorphisms, just very small individual amino acid changes in some of these bitter receptors are associated with particular types of pathologies. For example, sinus infections. That if you look at a big population of people who have chronic sinus infections, you will see a higher proportion of particular polymorphisms in the bitter receptor genes.
Interesting.
For the genes that are expressed in the airway and have been demonstrated to, this is work from Noam Cohen, as well as a number of other people at UPenn, but it's just fascinating because that's a real life example of, and now you're talking about people in a whole variety of different situations, different genetic backgrounds, and challenges abound in daily life. And when you just look at who suffers from a higher incidence of, so there are things like that. And there are many such examples, not just with sinusitis,
Yeah, a colleague of ours here at University of Miami Med School, in fact, is looking at the bitter taste receptor expression in tumors, oral cancer,
Huh.
and finding that they may play a role in carcinogenesis or.
Huh.
Well, actually, in the internal defense
Yeah,
against
tumors.
tumor cells.
But it's not just the bitter receptors as well. I mean, the so-called sweet receptors are also widely expressed throughout the body, and they've received substantially less attention as to what is going on with them. But it is a fascinating.
Right.
But they're seen on pancreas cells. They're seen in the intestine, and they're used for signaling to the enteric nervous system, for example.
Wow.
So they're not taking buds, and there's certainly no perception involved.
Yeah, that's an amazing story how you start out with, just looking at the specific cells in the tongue of a mud puppy, and then, you do the molecular biology, and, somebody finds the genes for the receptors, and then you find they're elsewhere and you don't know what they're doing. So there's a lot out there to discover. So in that regard, and this is directed at both of you, what are your projects that you're working on now that you think are the most exciting?
Well, one of the things that I'm quite intrigued by is the importance and need to have molecularly defined circuitry for taste, because that's how you can actually target interventions, right? So if you can molecularly define, okay, these cells connect to these other cells, and how do we know about dopamine circuits? It's because people, neuroanatomists, and developed use available antibody tools and other tools to say, look, these are the circuits that consistently get activated in certain situations. And similarly, we need to understand exactly which parts of these taste circuits, and how can you define the parts of the circuit that lead to perception, the parts that lead to reflexes, like the cephalic phase insulin release that we were talking about, just when you taste sweet. There are metabolic regulation parts of the circuit that aren't directly involved in perception, but they inform hypothalamic centers about nutrient content and nutrient density. So there are all these different circuits that proceed and diverge once you get from the oral cavity to the brainstem, nucleus of the solitary tract. And then after that, we have no handle on exactly which neurons speak to which centers. And I think defining that is really important. That's what.
Yeah, it's a huge technological challenge. I can imagine you're going to have to combine, say, optogenetics with imaging, and
Right.
Yeah, it's really complicated. Okay, Steve. What?
Well, as you probably recognize and realize that Nirupa and I collaborate, so we're working on the same problem. I use my skills in this functional imaging, the calcium imaging, using these genetically encoded fluorescent dyes, as we talked about. And we're also working on a problem of how is sweet sensed. I mean, the sweet taste receptors have been defined, of course, but it's looking like there may be other mechanisms for responding to sugars, specifically sugars like glucose. They may have a slightly different mechanism or additional mechanisms to the more like sucrose and other sugars. So sugar sensing and genetically identifying neuronal pathways using molecular neurobiology and functional imaging. I'd say that's what we're focusing on right now.
Yeah. Okay, it's great. Well, it's good to see you too, and talk tongues with you. I'm glad that you're getting support. I assume you're still getting support for your work. funding's not too bad right now, right?
Well,
It's always a
it's,
challenge.
It's
always
always-
a challenge, yeah, but,
Yeah,
we've done well.
yeah, good. Okay,
Matt, it's good chatting with you.
yeah, it's
fun
great to talk to
see
you
that
too, and
after all.
I hope the rest of your 23 is good, and yeah, it's really good. And since I retired, this is one of the things I'm doing, and it's actually a chance for me to have fun talking to people at the top of their field, and in this case, two people
Thank you.
I knew, have known since
Yeah.
since
Thank
the
you.
1980s.
Thanks, Matt.
Yeah, Matt, if, as you're going through and editing and redoing any parts of it, if you have follow-up questions or need to clarify something, just let us know.
And
Okay.
we encourage your listeners, if they have questions, look us up on the internet and send us your questions. We'll be glad to answer anything that might.
And I'll in the discussion, in the description section, below the video, and actually they also have this on the audio portion. I'm going to put links to your labs and also to a couple, your Nature Reviews Neuroscience review
You
article.
can
And
put in a link to the Purvey's textbook. The next edition will have a big chapter on taste.
Written by Nirupa.
oh, nice, good. Okay, it's great to see you. Take
All
care.
right. See you, Matt.
Bye-bye.
Bye.